Okay, I told you about the sheet. Welcome everyone, and I'm going to forego the business meeting until after our presenter gives us her program. So here's Jennifer. She's going to introduce her.
Hello, everyone. What a wonderful turnout for a summer meeting. I'm excited to see everyone here tonight. I noticed that the back is popular for everyone to sit in. We do have seats closer to our speaker. And our speaker tonight is Catarina Miranda. So she is a trainee with TMN, and she is also a graduate student working on her master's in biology at UTRGV. So if you're here last month, we said we were going to be talking about foraging, but Catarina is going to talk to us about her research on thorn forest canopies. So I'm really excited to learn about what she has been working on. Here's Catarina.
Hi guys, so like Jennifer said, I'm a grad student at UTRGV. Just to talk a little bit more about that, I'm under Dr. Bradley Christoffersen campus. Our lab is really small, but almost all of us work on thorn forest ecology and also a little bit on like germination of plants. But right now we're really going in the direction of restoration ecology because the lab is partnered with American Forests and U.S. Fish and Wildlife, and American Forests does the restoration down here now, and so that's what we're focusing on. And so that's what my presentation is going to be on. Am I in a good spot, or should I stand over here so everybody could see? Out of here?
People at home.
Okay. Is there like a clicker?
I'm just plugging that in.
Okay, thank you. So my presentation is called How Does a Forest Sustain Itself? And then there's a really long subtitle because that's how people make titles for research papers now. There's like a catchy short part and the really long part on what your paper is actually about. So this is going to be like my master's thesis. I just started my master's this past semester, so I haven't gotten into the writing stage. As I go through this, you're going to see that my results section says preliminary results. That's because we haven't actually like analyzed any of the data yet because I'm still out collecting a bunch of stuff all the time. I have field work 3 days a week for the entire summer. And yes, it's very hot outside, but it's fun. I don't think the clicker's working.
All right, you're trying to.
Okay, so I'm going to talk about a little bit of background, and I included this Texas horned lizard that I found the other day at one of my field sites, because one of the main reasons for this research is conservation. I'm kind of doing it out of order, but that's fine. So the various statistics out there for this are that there's only about 5 to 10% of native habitat left in the Rio Grande Valley that hasn't been destroyed yet. Of course, if you were to include all of the reforested areas, areas of like natural secondary succession, then there would be more. But I'm talking about like primary forest, virgin habitat that's never been touched before. There's very little left, as I'm sure most of you guys already know. And so a lot of our research on ecology and environmental science, that kind of thing that happens at UTRGV, a big motivation is because of conservation. And the horned lizard is not the most common abundant species nowadays, so finding this guy was really cool. So another reason why I'm doing this is for just like simple science reasons. RGV ecosystems are really understudied. There's only like a handful of papers that are on like thorn forest ecology, while there is an entire wealth of research on like tropical forests and temperate forests. There's very little on thorn forests, and some of that research does kind of transcend biomes and you can apply it everywhere, but a lot of it doesn't. And so it helps to just learn more about how semi-arid ecosystems around the world work, especially because semi-arid areas like this one actually make up some of the largest percentage of like land mass on the planet, even though everybody kind of ignores these places because they don't look as immediately impressive as like tropical forests or deciduous forests, stuff like that. So another reason for like this background is that there's been reforestation efforts going on in the valley for the past 3-ish decades. They started off being led by U.S. Fish and Wildlife Services, and now they're being taken over by American forests. But the way that this reforestation has historically been done is they go out somewhere, plant all the plants, or throw all the seeds out, and then they say bye and never come back. And so we want to know how these places are doing. How are old plots doing? What does restoration success look like? We know that probably reforested plots are not going to look like primary forest, like old growth thorn scrub, at least not for like at least 100 or 200 years. But what does success look like in these areas? And the other thing is that we want to know what are some best practices for thorn forest restoration. A while back, I was talking to somebody that worked for U.S. Fish and Wildlife out at Laguna Atascosa, and he talked about how there's no prescription for RGV restoration yet, that it hasn't been done long enough, and so we don't know like exactly what methods work. So we're still in the phase of whenever we do this research, we want to see like what works to begin with and what doesn't work. And so this kind of goes into my specific research question, which we'll get to in a little bit, but how can we compare reforested tracts to old growth forests? We have to find like different things that we can use to kind of like compare these two things. And the thing that I went with, or that we went with, because there's multiple people that are working on this, is canopy cover. If you go into a thorn forest in the valley, there is always canopy cover. It's a ubiquitous trait that you find regardless of where you are. You're not going to go into thorn scrub and there's just like bare open area for as long as like you see around you. So my research question is, how do canopy cover and forest successional stage shape the woody understory and its dynamics within thorn forests? That's kind of a mouthful. I'll go into it in a little bit. But there are also some sub-questions. So I talk about canopy cover. Canopy is just like the tree that's above you, the canopy of its leaves, and the shade that it's creating around that area. But obviously there's a bunch of different trees that grow here, so there's a lot of different canopies that we're talking about. Two of the most common ones, regardless of where you are in the valley, are ebony trees and mesquite trees. And we want to know, do these tree canopies have the same function? So whatever function they have in the ecosystem, is it just based on canopy versus no canopy? Or is there a little bit of species-specific stuff going on between these two different plants? Another one is the impact of forest successional stage. That just has to do with how old the habitat is. So is it old growth? Is it reforested? If it was reforested, how recently was it reforested? That kind of thing. And then lastly, we also want to look at if there are any species-specific differences in understory plants. So whenever I'm looking at the woody understory underneath the canopy of a tree, there's a host of different species that all like hang out there, usually at least 10 different species of plants. And are all of them interacting with that canopy in the same way? Or do some of them just kind of grow anywhere? We know that some plants in the valley, some woody shrubs, are super weedy, and you kind of find them everywhere. You find them underneath the shade of trees. You find them out in full sun. Others are a little bit pickier about the microhabitat that they live in, and we want to try and tease that out. So my hypothesis is split into two different pieces. The first one is that the woody understory will be denser and more diverse under canopy cover versus no canopy cover. That means that there's going to be more plants within the understory and more species of plants underneath canopy cover versus in areas of full sun. And again, like I haven't proved it yet, but this is what we think is going to happen. And we think that it's because there's a facilitative effect that happens due to nurse plant functions that are performed by large canopy-forming nitrogen-fixing species like ebony trees and mesquite trees. The second part to this hypothesis is very similar to the first one, but it has to do with the age of the forest instead of canopy cover. So we think that the woody understory will be denser and more diverse in older forests compared to younger ones. And this is likely because of lowered stress from invasive plant presence, herbivory, and disturbed soil conditions, which are all of the stressors that you would find in a recently reforested area. Okay, so I'm going to explain some key terms because I know a lot of this stuff is like science jargon. It's a mouthful. It just goes by really, really quickly. So what do I mean by canopy cover? Shade from a tree above. We know that most trees in the valley don't get higher than like 30 feet. And if you're out on the western portion of the valley, it's even shorter than that, like 10, 15 feet. But if there's shade from the thing that's above you, that's a canopy. We're counting that as a canopy. What is the woody understory? So this is woody plants in the forest understory, which is the lowest level of vegetation in the forest above the ground level. So the understory is like this tall. What is forest successional stage? This is the age of a forest from old growth to only 30 years post-planting. That is the youngest forest that we're looking at because it looks like that is about the amount of time that it takes for reforested plots to start to resemble the natural ecosystems of this area. And what is nitrogen fixation? So certain species of legumes, which are plants in the bean family, Fabaceae, fix atmospheric nitrogen with their roots via a symbiotic relationship with bacteria, and they increase the soil nutrients over time. And the way that they do this is all of that nitrogen that they take up through their roots, they accumulate it throughout all of the biomass of the plant. So the leaves have more nitrogen than like an ash tree or an oak tree or something like that. And then the tree will drop its leaves over time. If you've been underneath an ebony canopy, I'm sure you've noticed it looks like mulch underneath it with all the duff from the leaves. And all those leaves are super rich in nitrogen. Okay, so now I'm going to get into how I'm doing this research. So I'm going to start with my fieldwork locations. So the majority of my work is done outside. There's very, very little stuff that's done inside. I don't really work in like a lab with chemicals and stuff like that. I work more in like a field laboratory. I have two main locations. Those are Sal del Rey and Schleben Tract, which is along Highway 186. It's only a couple miles east of Sal. And there's three different types of forest successional stages that we're focusing on. One is old growth, as you can see right up there. Another one we're calling dense replanted is right here. This location is at Sal del Rey, and our dense replanted area is actually a place that was reforested in 1993, and it was done through a mixture of direct seeding and transplant. And as we can see, this is super, super dense. And then we have our sparse replanted. This is actually in this same location at Sal del Rey, and this was done only through transplants, and we believe that the density of the planting was about 100 trees per acre. So you could see that there are these really large areas that there was die-off from the stuff that they first planted, compared to this area, even though these are only 2 years apart. This one was planted in 1995, and those 2 years are not the difference between the level of growth. The method that they use to reforest the area is why these two areas look so different. And then our old growth forest is the Schleben Tract, which I mentioned is just a couple miles east of Sal along 186. So now I have different types of plots. So earlier I was talking about the three successional stages. And so these are, large like habitats. But within those large habitats, we are looking at a lot smaller microhabitats. And so the three different microhabitats that I have are called thick overstory, which is underneath an ebony canopy. So this is one that's underneath the canopy. Another one is thin overstory, which is underneath the mesquite canopy. This is also an area that has canopy cover. And then we have our full sun areas. And I know these two are a little hard to see. Whenever you're in one of those forests, it's kind of hard to capture the ebony tree all the way from the top all the way to the bottom because you can't get super far and everything's really dense. But in this area, you could see that there's no canopy cover here. Luckily, it was cloudy. It was one of those really nice days earlier in the season, but there's no canopy cover here. This is all full sun. And the reason that we call these thick overstory versus thin overstory is, again, imagine being underneath an ebony canopy. That canopy is super, super dense. All the leaves are super like packed close together, and you're getting almost full shade under there. Versus if you stand underneath a mesquite canopy, that canopy is very wispy and open. There's a lot more space in between the leaves and the branches. So there are differences in the amount of light that gets through both different types of canopies. And so for this study, the plots have to be replicated for a proper sample, and any ecological study would be like this. But because the plots have to be replicated, that means that I have multiples of them. So one of each plot side by side is, we're calling a block. So one ebony canopy, one mesquite canopy, and one open area all together in kind of like a little triangle is one block. And so at each of my locations, I have 3 blocks. That means that I have 27 plots total. And a lot of these plots are really big. Some of them are like 10 by 10 meters. So it's taking a very long time to go through them. So what is it that I actually do whenever I'm out there? So we're doing plant surveys. So within every plot, every woody plant that's less than 50 centimeters tall is measured and tagged. As you can see here, this is our ebony canopy, or our ebony trunk. And then there's a bunch of tags around here. These are all of the plants. And that was before I even finished this plot. And then here's my little tape going through, because we have a coordinate system that we use. So the data that I collect is the plant species. So we find a plant, it's under 50 centimeters. First thing is, what plant is this? Well, let's say it's a guayacan. And then the next thing is we take coordinates within the plot. So every plot that we make, it's either a square or a rectangle, and we set up these super long meter tapes so that our plot has like an X and a Y axis, and then we use that to make a coordinate system. And then we measure the height and the crown of the plant. The crown is like a measurement that goes this way around the plant, because a lot of our thorn scrub species are very bushy. They don't just grow up. And so you want to get an idea of how much space this plant takes up in general. And then we also take stem diameter, usually at ground or as close as we can get to that. And then we also measure mid-story presence and or composition. This is something that the measurements that I've been taking have changed over time. Right now I'm just doing mid-story presence, like yes or no. Before, I've done a lot more detail. So what are some of the preliminary results for this study? So there is natural forest regeneration that occurs even in sparse landscapes. And I included this picture because right here, this is all invasive grass. And whenever you go out and look at that, it doesn't look very nice. Seeing the grass can be a little bit depressing, but right behind the grass, there's an ebony tree right here. There's an ebony tree right here, and then there's a bunch of mesquites behind there. And I know the picture quality isn't the best, because if I took my fancy camera out to my field sites, I would never get any work done. But there's a full understory and midstory of plants beneath those large trees. And so they're serving as sites for regeneration. The other thing is that microhabitat means everything. And by microhabitat, I mean like underneath the canopy versus not underneath the canopy, underneath an ebony tree versus underneath a mesquite tree. It really means everything. There are way more seedlings underneath the canopies versus underneath full sun. And in this little graph that I made, you could see that like the seedling distribution in these 3 different microhabitats. So seedlings underneath thick overstory, which is underneath our ebony trees, is the bar in blue. Underneath a thin overstory or the mesquite tree is the bar in green. And then out in full sun in the open areas is the bar in orange. And you could see that the majority of the seedlings that we've measured are occurring underneath these canopies, not out in the open. Now the data is going to look very different once we're actually done with it. This is after only measuring like, I think maybe like 350 seedlings. Now I'm almost at 1,000. So it's going to look very different, but I can tell you even anecdotally, there is way, way, way more stuff going on underneath the canopies than out in the open. And something that I didn't include here is that there are kind of some species-specific preferences for canopy versus no canopy between the different plants that we're looking at. And this is something that, again, is going to come later in my study because we have to analyze a lot of data for this. But one of the plants that we look at a lot that we see out there is snake eyes, the one with the fruits that are translucent with the little black seeds in the middle. I've never, ever seen one of those in any of my open areas. And I'm already done with, I think, 4 blocks. So that means that I've already done 12 different plots. I've measured over 800 seedlings, and I've yet to find a snake eyes out in full sun. And that's something that's important because whenever American forests and US Fish and Wildlife and anybody is doing these reforestation projects, if you know that a species like snake eyes, which we know is important in thorn scrub because we see it everywhere, it's likely functioning as some sort of keystone species out there. We know that it's important, so we want to include it in these plantings. But if we know it's going to die if we put it in full sun, then don't plant it out there at the beginning. That's something that we want to look at for best practices of reforestation. Just checking the time. Okay, so this presentation is kind of in two portions. I wanted to start off with my science because I'd never presented on my research before, but this is a group of a bunch of naturalists, and I know what we all get excited about. So the second half of this presentation is about all the cool stuff that I see out in the field that gets me super excited. And again, to me, it's really important to have this blend of like science and being a naturalist. I think like purely being a scientist and never having any of that like wonder and amazement as you go through nature is so boring. I don't want to live that way. Like I want to learn how to do science, how to be a good scientist, but I also want to hang out with Texas horned lizards. So this is a horned lizard that I saw at the Ituria Ranch out in Willacy County. There's a reforested tract there that we're monitoring, and a lot of the plants are looking really, really bad. I went out there to like mark some transects and look at survivorship of plants that are out there in this area that was planted just a couple months ago in October of last year. And these plots are really, really big. There's several acres. And so you kind of have to like drive around the perimeter to be able to get to all the different sides that you need to get to. And I was out there with the person that accompanies me all the time because I don't go out there by myself. I have an undergraduate research assistant. And like we get out of the truck and we start walking and I see all these harvester ant mounds and I say, this is prime Texas horned lizard habitat. And then we get going and we start measuring, or not measuring plants, but like marking down survivorship for plants. And she's like, oh my God, look down. And there's a horned lizard right there. It was so cool. And they're so nice. And they're also really slow when they run away compared to like the whiptails and stuff like that. They're so slow. We see lots of Texas tortoises out at our different sites. This one I think is at Sal, but I also see a lot at Schleben. They're so cool. This is a rattlesnake that I saw. This is the biggest one that I've seen yet. I've only seen two, and this one, I was actually in a very scary situation when I saw this one because I didn't see it until it was right next to me, but luckily it was asleep. I don't think it had any idea I was there. It wasn't sticking its tongue out or anything. And seeing them can be a little bit scary, but it's also really exciting. I love seeing them. This is a Texas tan tarantula. So there's one location that I'm doing some work at that I haven't mentioned yet, and that's the Martin Refuge that I think a lot of you guys might have been to before. I know one of the field trips that I went on with the TMNs was to the Martin Refuge. I haven't mentioned it yet because I haven't actually started measuring anything there. I just went out a couple weeks ago and set up some temporary flagging to mark where my plots are going to be. And then probably next week, we're going to go out and actually like set up our giant blocks to figure out like where stuff is going to be. But we were out there the other week and this guy came out. This is a rhabdotis snail and a Yucca trichuliana flower. I see the rhabdotis snail shells everywhere, like in my yard and stuff. Whenever I'm planting stuff back there and I start digging, there will be rhabdotis snails in the soil. I hadn't seen one alive, and here they're thriving. This is at Sal del Rey, and every morning, if it's like dewy enough, they're out, just out and about doing their thing. They're super cool. This is a type of yucca moth. So I don't know if you guys know the relationship between yuccas and yucca moths, where the yucca flowers are only pollinated by yucca moths. They have this really specific relationship between them. It's really cool. This is one called a bogus yucca moth. I'm not sure why it's called that, but it's super tiny. And I found it out on a day where it had been raining, I think the previous day. That was really cool. This is a pipevine swallowtail. You might notice the significant increase in picture quality for this one picture. That's because every time that I was going out to Sal for field work, I noticed this really gorgeous wildflower patch along 281. And every time that Haley and I would pass down, I would say, I need to bring my camera next time so we could stop whenever we're done and photograph everything and collect seeds and just do all of that. And it took me forever, but I finally remembered to bring my camera. And we stopped at this like random RV park and got out and crossed 281 to get to the median. And there were pipevine swallowtails everywhere. It was amazing. There were so many of them. That was really cool. But like I mentioned earlier, I cannot bring my camera out with me to do field work or I would never get anything done. So very few beautiful pictures. But this is another one there. There were also lots of swallowtail caterpillars. The caterpillars of this species here are really cool because if you go like just a little bit up north, like San Antonio, the caterpillars are black and the red parts are just the little tubercles right here. But in the valley, they're all red. And there's actually a paper on this that's pretty easy to find on Google Scholar. It might even be open access, but they did an experiment to see if the color could be brought on via the climate. And so they raised pipevine swallowtail caterpillars in different temperatures. And sure enough, the ones that were raised in hot temperatures were all red, and the ones that were raised in lower temperatures were all black. And it was really consistent. So it's probably not like an ecotype difference. It's more just something that happens as some kind of like phenotypic plasticity that happens as a result of the temperature. This is a Theona checker spot. I think their host plant is Senecio, right? Yeah. And I think it's on a Senecio right there. I've never seen one at home, even though I have a bunch of Senecios, but they're super pretty. This is a Texas wasp moth. I thought that was so cool because it looks like a wasp, but it's not. And the only way you can kind of tell from afar is just looking at things like this, like the legs and like the antenna. Like it looks like a wasp, but there's just something off about it. And I had already heard about this species. And so whenever I first saw it from afar and I was like, is that what I think it is? That's not behaving like a wasp. So I got closer and sure enough, it was a moth. That was at the Schleben Tract. The Schleben Tract is relatively small, but there's a lot of diversity there of animals and stuff. If you trek through there and look at the plants, there's a surprisingly small amount of plant diversity in Schleben, which actually makes us think that this tract is not as old growth as we think it is. It was definitely not reforested, but it might only be like 100 years old, because whenever we go to places like the Martin Refuge, which are not that far from Schleben, the diversity of plants in the understory at Martin Refuge is crazy high. There's so much there. There's new stuff everywhere that you look. Whereas at Schleben, the host of species is all pretty similar. So one of our theories for that area is that it was cleared maybe in the late 1800s, early 1900s, but it started the natural succession process again prior to a bunch of the invasive species that we have here now being introduced, like the different grasses and stuff. And so that's why you see places like that and they look pristine on the inside, but they don't necessarily have all of the characteristics of old growth forests, like there being a lot of dead wood everywhere. There's really not very much dead wood out at Schleben. And there's also not a huge amount of plant diversity, but insects and stuff like that, there's a ton of that stuff there. This is a California warrior beetle. These are all at Sal del Rey. If you're paying attention to the ground while you're walking around, you'll find these. And actually, me and Haley, my research assistant, we have lunch in the field every day because it'll take too much time to walk to the truck. And if you sit down in those areas for long enough without moving, these things come out and they start to walk around super, super close to you. And if you move, they freak out and they sprint in the other direction. It's very cute. This one was really upset about me holding it, but I wanted to get like a good diagnostic picture for iNaturalist. This is a velvet ant. We see a ton of them out at Schleben. They're really cool. Joseph, do you know if this is a female, right? The males look very different.
Yeah, the males will look more like a wasp, so that they have wings.
Okay. Yeah, I never see any of those out. I always see the females out. This is palm leaf globe mallow. This is in the family Malvaceae. This is more for like the plant nerds like me out there. Whenever I saw this plant out at Sal, I was so excited. I'd never seen a globe mallow before. I'd only seen it in the pictures of plants of deep South Texas, and it was so exciting for me to finally see this plant. This is brushland lantana, one of the different lantana species. We have a lot of lantana species down here, surprisingly. Again, this is one that is not super common in cultivation, but if you go to like native plant gardens at people's houses, I feel like you can usually find this species there. I'd never seen it anywhere in habitat, but at Schleben, it's everywhere in the understory. This is two-leaved senna. If you walk along the trail at La Salle del Rey, you're going to see a ton of these. They're just lining the way everywhere. They're very pretty. This is pencil cactus, the Cainocereus pusselgeri. This plant is so exciting to me. I found this at the Martin Refuge, and the way that I found it is probably the way that most people who know this plant find it, which is like you're walking around looking for something else, and then you find that, and you're like, oh my God, this is so cool. And of course, I got super, super excited over it. And there was actually 2 different individuals of this in a similar area. So there's probably an entire population out there, which I find super, super exciting because you really don't see this plant super often in a lot of places. This is Chomonke, Nawatlia hypaluca. Same thing with this one. There's one that's right along the trail that Audrey showed me whenever we were going on a little tour of the area. And I got super excited for that one. But this one I found on my own, which I got even more excited for because I thought that there was only one of those in the refuge, but there's, again, an entire population of this plant. This is an Asteraceae that's a tree, which is super cool. I think it's the only one in the valley that's a tree Asteraceae. Super cool. And the undersides of the leaves are like this silvery white color. It's a gorgeous plant. This is an all-thorn, but it's a baby, and I think it's super cute. I called it cute, and Audrey Martin laughed and was like, I've never heard anybody call all-thorn cute, but actually my advisor, Dr. Christoffersen, he made a joke and said that whenever you walk past all-thorn and it like pokes you, it's not that bad. It kind of feels more like acupuncture than anything else. At least it's not curved like a lima. So this is desert yaupon, Schaeferia cuneifolia, and it's not a super rare plant, but the reason I included this picture is because of this orange. Something that I discovered, the orange along here, is that every desert yaupon has that orange staining that if you scrape the trunk of it just a little bit, that orange starts to come out. And so before I got really good at IDing this species just from the leaves, I would use that. Like I would see the plant out in the field and I would say, I think that's a desert yaupon, but I'm not sure. Let me scrape the trunk just to make sure. And sure enough, every time it was orange. And I have no idea what causes that. I haven't read about it anywhere, but I thought that was really cool. This is an ephedra species. This is one I also got super excited about. This is one of the only gymnosperms that we have in the valley. The one that everybody knows is a Montezuma cypress. This is also a gymnosperm. These are cones. Super crazy. And it's also a dioecious plant. So this one is a female plant. However, I don't know which ephedra species it is. I can't tell if it's Antisyphilitica or pedunculata. Hopefully somebody here can help me with that. But I actually got one to germinate in my garden. This is I think it's a miniature barrel cactus, Hematocactus hematocanthus. And this is actually out at Laguna Rascosa, the Delaney tract, which I'm going to be doing some work in monitoring some reforestations. This one is just right along a levee that's in between like 2 areas of Arasaka. And it was just right there. That thing has to be like decades and decades old. These are earth star mushrooms. I'd never seen anything like this before, but if you go hang out in any of these tracks, and this is actually at La Salle del Rey, which again is reforested. It's not old growth, like super prime healthy habitat, but obviously there's something going on there that's allowing for cool mushrooms like this to come up because I do not get these in my yard. I have a ton of native plants there. And there's also, you could see on the side, some of the lichens as well, growing right here and right up there also. There's a bunch of cool stuff that's going on in the soil out in those areas. I'd never seen those mushrooms before. They're so cool. I believe that's everything. Yes, my acknowledgements. I'm funded by American Forests. I'm supported by the Christoffersen lab, which again is a really small lab, but it's super cute. It's super cool. There's a lot of stuff that's going on there that we're doing. And at some point later on in my research, there might actually be an opportunity for volunteers to get involved. And so if I do manage to figure out an opportunity for that, I can definitely let you guys know. Because it's always great to get more people out in the field participating in research, and it'll make my life easier if I don't have to go and resurvey. There's probably going to be like 2,000 seedlings by the time I'm done with this thing. So if I don't have to go and resurvey them all at the same time a year from now by myself, that'll be amazing. And yeah, that's everything. Contact info, my professor's contact info. Thank you. Yes?
Talking about these things having been planted, canopy trees, and but that the undergrowth stuff doesn't grow unless they're shaded. So that means that you have to go back once they're grown and replant.
A little bit, yeah. So there actually is this concept that is used in reforestation in other parts of the world called underplanting, where they go through, do the initial reforestation, come back 30 years later and do reforestation part 2 as the underplanting portion. And for that one, it's more for diversity and also to build up the understory, because a lot of reforested areas, if you just plant them and leave them alone, then they end up looking kind of like a park where there's a bunch of canopies everywhere and then just grass and that's it. So yeah, that's called underplanting. I think it'd be really cool if they tried it here, but no one seems very interested yet. Maybe after I publish my research, they will be interested. Yes?
You have a list of the native plants that are big ebony trees.
Not yet, but I can provide it just anecdotally. Does anybody else have any questions? Yes?
Catarina, I'm online. I don't know if you can hear me.
Sorry, 1 second. Somebody in person is asking a question.
Oh, sure.
You mentioned in your studies primarily ebony and mesquite. Are there other species of taller growth like an aqua or something of that sort?
No, there's not. There's several reasons for that. So if I added in more species, the amount of plots that I have would kind of increase exponentially, and I can't do that. But the other thing is, because we're at the UTRGV Edinburgh campus, we do tend to gravitate more towards field sites that are close to us. And trees like anaqua don't really grow naturally out in like northern Hidalgo County or like western Hidalgo County. They tend to be more eastern species or like delta species. So a lot of those other really big trees like anaqua, cedar, elm, Montezuma cypress, they likely do serve a similar function, but they don't occur in any of my field sites. So we don't study them. The other one that gets real big out at my sites is goma, but within our blocks, everything has to be relatively close together. They can't be super spaced apart, and it's difficult to find a place, or at least it was, where there's an ebony and a mesquite and an open area and a goma all right next to each other. So it's easiest to just stick with those two, but also in northern Hidalgo County, western Hidalgo County, Western Valley in general, the two biggest trees really are ebony and mesquite. And then of course there's blackbrush as you get further west, but it doesn't get as big as the ebonies do. I think someone online had a question?
No, you've answered it. Thank you.
Oh, okay. Does anybody else have any questions? Yeah.
Like anecdotally, just based off of what I'm just seeing out there, are there differences in where under canopy they seedlings tend to grow? So I know Brad had mentioned drip line stuff.
Yeah, there definitely is. So if I were to like draw a picture with a bunch of little dots of where most of the plants are, you would see that the majority are clustered a couple feet from the trunk. and then there's less as you get further out towards the drip line. So the deeper the shade is, the more seedlings there tends to be, or plants in general, because they really are not seedlings. A lot of them are juvenile plants are already at reproductive maturity. But yeah, there is a difference between that. That's part of why we have the coordinate system going. Part of it is so that we can find plants again later. But the other part is that if we have this coordinate system and we know the dimensions of our plot, we could potentially draw a little diagram of every single plot that we have. So you could see like a little map of where all the different plants are. Any other questions? Yes.
Go ahead, John.
Yes.
Couple of comments. First of all, your enthusiasm is just awesome.
Thank you.
love it. And secondly, while I know you haven't specialized academically for a career, your being and intending to remain a generalist is very impressive to me. I think that's a great thing. To call yourself a master naturalist, you should be a naturalist across the board.
Thank you. Well, it's how I started, so I don't want to depart from that. Like, it's what got me excited about all this stuff to begin with.
That's great.
I think Anne had a question.
I was gonna ask you what your plans were for the future. You're studying all this, towards what goal?
I don't know. Everybody asks me that all the time. I don't know. Something in the field, but I don't know exactly what. I haven't made up my mind yet.
Another question over here. You gotta ask another question. We have to get closer to quarter after. Advanced training hours. I'm always careful of that. Okay, another question. Come on. Did you have one here? Oh, we got one in the back.
Surely rain must be one of your variables. Are you monitoring as well?
We have not, but we can because of like weather stations and stuff like that. We'll be able to do that. But because we're not really including very much of a temporal aspect to the study, things like weather events aren't super important to the design. But yeah, I think that we'll have access to rain data, like kind of a little historically, so we can use that if we need to.
I have a question. Go ahead,
Yes.
Bill.
Are you gonna be able to expand it? Because obviously you're gonna have other critters all the way up into the mammals that have a lot of effect on this.
Mhm.
You talk about the ebony tree, the creatures that eat the ebony pods.
Yeah, so including herbivory in the study would add in too many variables. And one of the things that my professor says that his role constantly is narrowing down the students' interests because we want to do everything. And it would end up being like this gigantic study that is not within the like realm of a master's thesis. So no, we can't really look at herbivory because of that. Yes.
So I know you said that you would share like a list like later maybe, but what are some of the species you found underneath the ebony tree? Because I have like a really big one at my house, and then we got like a smaller one at my dad planted near there because he was like, they can connect, you know?
Yeah.
You know they will. So I'm just curious to see like what else you think might be.
Yes. Okay. There's guayacán, there's snake eyes, there's goma, granjeno, Brazil, coyotillo. Those are the main really common ones. Out at Sal, there's a lot of cat claw acacia because that's one of the species that they planted a lot of at the reforestations. So there's that one. Desert yaupan, allthorn. There's way more, but I would have to look at my data for that. It just slips my mind.
So do you think it would be smart to try to like find those different species and plant them near there then? Or
Like for your yard?
like,
Yeah, if you'd like to, yeah. I don't know how most people like to do their native scaping at home. I still live with my parents and they let me do almost whatever I want to the yard, except my mom is like, you cannot cover the house. The house is pretty. I want it to be seen from the street. So that means in the front, I can't have like a forest. That can only be towards the back and on the sides. So I haven't done something like that yet. But if you wanted to create like a tiny, tiny, tiny forest in your yard, then you could definitely do that. and the plants will be just fine.
Yeah, because we have some like pollinator plants that I got planted there. And then we do have some natural forests that was just there already on the property, like some Saint palm, some Barbados cherry that's growing there.
Oh, that's
And
another
there's
one we
like-
find.
And there's like Foma that's growing there too.
Mhm.
And there's like aqua there.
Yeah, one thing for, I guess, like a more urban suburban setting is if you do create like that, like the layers of the forest, that tends to be better for bird habitat than like only trees or like only butterfly plants, because a lot of the birds here like to hang out in the mid-story. And so if you kind of like fill that up, you'll probably get more birds hanging out in the area also. I think someone in the back had a question. Yeah.
Yeah, well, for your next research. But on an NPR program a while back, they were talking, and this is more deciduous forest, where there's a relationship mainly underground between species, different species, where they're actually sharing nutrients and so forth. That might be an interesting thing for you to study with your dominant tree and then all the undergrowth. There's any cooperation.
That would be really, really cool to look at. I know nothing about underground plant interactions, so that would be a totally different field for me to go into, but that would be super interesting. And I wish that there were more people in the valley studying this stuff, because there's just so little.
We got time for just her question, your
I
question.
recommend you read The Mother Tree. Yes, this woman made her life studying the mycorrhizal relationships of fungi, transferring materials from one tree to another. It's just a fascinating
Yeah,
book.
the tree died and will transfer on.
Yeah, they talk to each other.
In my opinion, two things that really need to be looked into here that nobody has looked into is some of the soil biology. If you go out to a lot of these places right after a rain, the ground is completely alive with fungus, with lichens, with bacteria. You could see like nostoc, that weird bacteria that looks like seaweedy. And I don't think anybody has looked at that in the valley so far. There's got to be a huge diversity of organisms that live in the soil crust here. And it'd be really interesting to just, like, first of all, classify them. There's probably a bunch of undescribed species that are living here that nobody has studied before. But then also looking at how they function in the ecosystem. And then the other thing that I learned more recently is that everybody always says we have no idea what the valley looked like 500 years ago, before all of like the land use history that causes it to look the way it does now, before all the cattle came in and kind of like destroyed all the grasslands that were here. We don't know what was there. And something that I always hear is it's impossible to know. But then somebody told me about pollen studies that like paleobotanists can look at fossilized pollen in an area to characterize the vegetation, like the historic vegetation of areas. And I mean, from my really, really basic understanding of it, I think that it's possible to do a pollen study from pollen that's like 500 years old. I don't know if that's enough time to fossilize or if it would just be there hanging out. But for somebody to come down here and do that would be amazing. That'd be super cool. Because then we would know like what plants were growing here back then. Yes?
Studying ebony and mesquite,
Yes.
are native plants, but they developed 500 years ago. 500 years ago, there was zero mesquite in Texas.
How do we know that though?
Because mesquite came out with cattle from Mexico, and it's in the feces.
Mm-hmm.
That's how the mesquite got.
But can we definitively prove that there were no mesquite here at all as of right now? I mean, if with a pollen study, we'd be able to do that, but like maybe more of it came up. Oh, that's true. Yeah.
Okay, thank you so much, Catarina. We appreciate it. Right.
And we'll start our business part of the meeting.
Okay, this is starting the business section part of the meeting, and we'll go with the treasurer's report. Gail, our treasurer. We're waiting for Joseph to put it up on the screen. Okay, while he's working on that, I got a couple things to tell you. Let me see. I'll go over it at the end, but the speaker was 0.75 AT hours when you do your reporting. Okay, and I am going up north to see my family for about 6 weeks, so I won't be here for the general meeting. I can do all the board meetings and the agendas, but Roberta is going to, she's first secretary, president, vice president, and she will run the general meeting. So you won't see me here. It's not, I just didn't croak or something. So then I'd see you in August. So Gail, okay, here.
Okay, so we've got the report up there, just a little bit of activity during the summer months. We slow down. The main expenses were for the new class for the graduation, and so that was the most that we had. And then, of course, every month we pay for our facility usage. A little bit of sales came in just from miscellaneous items. That's it.
Okay. All right. Thank you, Gail. One other thing I want to mention while you're all here and you haven't started to disperse, I need somebody who would be like hospitality person. We have somebody for our classes all the time, but we don't have anybody for our chapter who would just come. I have a tote and it has paper plates, napkins, plastic forks and spoons, anything that we use to put out on the table. And I forgot it tonight. And the church's stuff is here. So now we're not supposed to use their stuff, but we've had to. So now we got to pay a heavy fine probably. So if somebody here, think about it, your main responsibility was taking that tote and bringing it home and bringing it back the next, and taking out the plates and the napkins and putting them on the table. That's the extent of your responsibility. If you're interested in it, stop. Don't everybody stand up at once. Please, someone help me. You've already volunteered. You hear that, Donna? Just, you're not here in person tonight and they're just throwing your name out constantly. Okay, now the minutes of the meeting, I sent it out to all of you. Did you see anything that you needed to change or you wanted to mention? Okay, then the minutes will be accepted and filed as Velva has written them. And now we got some directors and committee reports. Jennifer, you want to come up and talk to us about our next speaker?
Hi, everyone. I am working on lining up speakers for the rest of the year. So I don't have someone locked in for July, but I am working on it. And so if you know someone who you would like to have speak at one of our meetings, please let me know so that I can contact them and see if they're interested in coming. I do have some great leads from people already, so hopefully we'll have some very interesting programs for the rest of the year. Thank you.
Thank you, Jennifer. Okay, and our membership director, Jaime Rodriguez, he was not able to be here. Is he online? Oh, I asked him to send Joseph his report. So let me see, I think I have it here also. So I asked him to do number of members reporting, either AT or volunteer work. So for the month, let's do the month of June, 23, and it's way down. I don't know if it's because winter Texans are gone and it's too hot or people on vacation, but you can see in February, we had as high as 77 members reported hours. Now in June, we had 23 and different opportunities. Looked like February was a good month. In March, we had 32 as our high, different opportunities. In June, we only had 13. Volunteer service hours, oh my goodness, that really took a dip. Probably March was our highest total with 1,400 hours. And then in June, we had 155, 156 hours. And
June right now.
pardon? Oh, we didn't, oh, you're right, we haven't finished it yet. Okay, all right, I'll give you that. We're not finished, June. Get busy, get busy. Hurry those numbers up. Okay, and advanced training, wow. Okay, the high was March at 284 advanced training hours. I guess people were trying to get their rattlesnake pin. I think that was that. And now for June, 42. But by now, probably a lot of people have already gotten certified. So we got some people getting their rattlesnake pin tonight. Okay. Okay, that's about it for Jaime's report. And awards and recognition. Okay, I have a list here. I guess if I say Logan, we can get all of these taken care of. Logan, he got his initial certification, and he got his rattlesnake pin for this year, and he got his 100-hour pin. So come on up, Logan. Wait a minute. There you go. Thank you. There you go. Congratulations, Logan. You worked hard these last 2 months. Okay, we have a 250-hour milestone pin, Bill Rich. No, you'd reach that? Is this a surprise or you know?
No, I watched mine.
You were watching your hours. Okay, Liz Eddy, 100-hour milestone pin. Congratulations. And Janet Schofield. She got her rattlesnake again. Congratulations. Thank you. Thanks. Okay. That was one thing, the class, right? The students, this year's 2024 class, they love the rattlesnake pin. That was their goal. We got to work hard and get our rattlesnake pin. So hopefully the members are still working on it. Next, we got Anita. Is Anita here? Do you have anything to report or anything coming up you want to announce or? Okay, nothing for her to report. I know you could say, start working on your article for the next Chachalaca. Right. Yeah, I said it for you. Okay. Oh, yes, I had Anita, if she'd be interested in doing a workshop on writing. So people who are a little bit interested in writing, if you wanted to take her workshop, the idea would be to get more people to write articles for the Chachalaca. I know myself, for one, I've never gone to her workshop, but I think I'd like to because I have a lot of stories in my head.
Chock full of information, and you will leave just really wanting to like something.
She points to somebody who has taken her class and she's shaking her head no. Oh, okay. Is there anybody in this room who would like to take her workshop and learn how to write? Look at this. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We got 10 people. You got a class. Go ahead. Yes.
There was only 2 people from this
Chapter.
stories in the Choctaw. It is a joint. So it's the memberships opportunity.
To what? Brag? To brag about our doings?
So it's the memberships piece of literature. If we don't submit to it, it's going to go away.
Oh, we wouldn't want that, would we? Yeah. Okay. Yes.
Would that be against you? Yes.
Yes, advanced training. Good. I'm for that, right? I'm for that. Get your advanced training. Yeah. Yeah, set up a day and then tell who's advanced training, Rivers volunteering.
On the next chapter,
Judy, yeah, Judy.
fall issue.
Yeah, you tell Judy, she's in charge of advanced training. Okay, very good. Joseph, you have anything? Okay, Joseph has that. Hey, did you know those moths that the speaker was talking about? Were they?
I've heard about them, but well, the Texas wasp moth, yes, I see that frequently. The Yukon moth, or no, what it was, the bogus moth, I've never seen that, but I've heard that it's like the only thing that pollinates those flowers. Hey,
And do you only see them in the day, that moth?
I,
She saw them on the
maybe.
flowers. Maybe they're just a day thing instead of your night.
Yeah,
Your night.
maybe why I don't see them.
Okay, let's see. Judy, advanced training. You got something to report to us? Here, hang on. So wait a minute, hang on. So the people at home can hear, we're putting Judy.
I try to get something out near the beginning of every month. And then if I hear other things, I put those up as well. So if you know of something, please get it to me and I'll get it out to membership. But one of the things that I told Anita, if you can't get a hold of me and it's something that's going to happen right away, go ahead and get it out yourself. Or send it to her because, you know, the important thing is that it gets out to the membership. And I don't want something to happen and just because I'm out of whack, that the membership doesn't get a hold of it. I did get something today that I sent out. It's going to be in July, but I just wanted to mention it since I'm here tonight. This was sent to me by Robert Gaetano from the Rio Grande Valley chapter. And there'll be an open house of Rio Grande Valley Native Plant Restorations Operations Workshop. That's a mouthful, at the Marinoff Nursery in Alamo, and that will be July 12th. So that went out to you today, and I'll get it in my July list. But since I'm here, I wanted to mention, you know, look in your email because you should have that. We had a great field trip this last month. Bianca Banda, thanks to River and others, got us hooked up to go to that ranch, and it was really absolutely amazing. And for the month of July, where's Joseph?
I'm over here.
Oh, there's Joseph. I'm going to turn this over to Joseph because he and Donna are going to do something for us in July with bats and moths.
Suggestion?
Yes.
Tell people how they can email to the group.
Okay, on our bat field, we're talking with Donna about a bat field trip. I usually, Javier leads it. He's not available this year because he's got a baby at home. So I'm going to lead it, and Donna is going to help out and bring the equipment that from the big state project that we're using and talk to us about that too. We're at least tentatively planning. We're looking at the next 2 Saturdays, so it would be this month instead of July, although with the weather, that may not happen. So we may end up moving it to early July. And then we'd meet out at the Conway Overpass in Mission again. So if you've never been there, there's a whole lot of bats living under that overpass. And they come out just a little bit before sunset. And we'll send you more email you details when that happens. If we did do it, if it doesn't rain and we did it this Saturday, is there anybody that wants to do it that early, or we need more notice? Raise your hand.
2, 3, 4, 5, 6, 7, 8.
This Saturday? Is that all right? Or how about next Saturday? Does that sound better? Okay, so we'll watch the weather.
Either or.
Yeah. If one of them looks better but for weather, we'll try that or plan for July. And then Moth Week is at the end, toward the end of July, the 20th to the 28th. There's going to be a bunch of Moth Week events. Quinta Mazatlan has a big one. They go till midnight. There's usually hundreds of people going through there. He's looking for volunteers to help out. So if you know anything about mothing, you might want to help. Or if you're really good with and fast with iNaturalist on your phone, you can make yourself look like an expert.
I've already signed up.
And so, wow. And Benson State Park is going to have one. Estero usually has one. I haven't got the dates on any of these yet. And we're going to do a chapter field trip at Oleander Acres, which I also haven't figured the date out yet. But all of those emails will come as soon as possible, and any other I hear of. So there should be plenty of chances for mothing from the 20th to 28th.
Right, this is
A brochure on bats, and it has 32 bats. Most of them we have. Do you know the exact number?
I think we have about 14 here. 14
bats here. This is a poster, and it's on. Check the website to see if it's still available. I think it's www.text. Yeah, it's a text brochure, and it looks like this. Texas bats. Yes, so check it out if you love bats. I love bats. Okay, where were they now? I did. River, do you have anything you'd like to talk about?
Hello. Okay, so for volunteering opportunities, we have Ferry Fest coming up on July 20th, I believe. Yes, July 20th. However, volunteer opportunities have already started because we have 4 more weekends before the 20th of July. And you can go to the World Birding Center in Edinburgh and just show up. Their hours, I have them here. My phone would stay on. Their hours on Saturday are 8 to 6, and on Sunday 12:30 to 6:30. So that's 8 in the morning to 6 p.m. on Saturday, and on Sunday from 12:30 p.m. to 6:30 p.m. So you're supposed to show up. Just tell them at the front desk that you're there to volunteer for the Ferry Fest, preparing decorations, etc. They'll tell you what to do. So 4 more weekends of that before the actual event. Then on the actual event, they will need people to man different booths that they're going to have. So you can arrange that while you're there volunteering to prepare the decorations if you like. Also, our chapter will have a table there, which probably Roberta is going to get people to man. That's what our president told me. You're on the spot.
Well, I'm gonna be in Ireland, so.
Oh, you won't be here? That's fine. I can do it. I can do it. If you're gonna be gone, I can do it. Not a problem.
If anybody wants to help River too.
Yeah. Yeah.
Get a hold of River and let her know that he'll work the table. It's just outreach, talking to people about our TMN chapter and what we do and giving information.
And given information about how to join our chapter through classes.
Yes, and we're hopefully gonna develop an activity that we can entice the kiddos to come and visit our table with. But I'm not like super duper creative. And so if you are volunteer, yay, Melissa, thank you. Excellent. I can do that, but I don't know if we'll have an activity, but if you can come up with an activity, it'll be great.
I think Donna's volunteering. She's got her hand.
Excellent. Thank you, Donna. Yes, as a matter of fact,
River, in the past, we've had the activity of the tattoos. The kids loved those tattoos. And Anne should have all that.
I passed them on to Roberta.
I'll get them from Roberta.
Yeah, they loved that.
And when you bring the kids over, they have to come over with their parents, and then those are the people we're trying to get interested in joining TMN.
Exactly. Okay, so again, the Fairy Fest is July the 20th, but volunteer opportunities for that have started on the weekends, and you just show up for that. The other one that's ongoing is Adopt a Loop, and several of us went and actually did the track, the Ituria Brush Tract and Benson State Park one day. And what you do is you go over to the sites, and all of this is an email that I sent out already, but I can send it out again. So you go to any of the loop sites. You don't have to go to all of them the same day. You can pick one and go on one day. And believe me, we were worn out by, I don't know, what, 11? It was like too hot. It got too hot too quick. But anyway, you go to the site and you take pictures of the vegetation and anything you find like that, wildlife, and you put it on iNaturalist. And you look for the project, Adopt a Loop, and it has a number. It's not all that hard to do. I mean, if I was able to find it, you all will find it, because I'm very technology challenged. So it's okay. You'll get it. The birds you would report on eBird, and everything else on iNaturalist. And then you count your drive time, and you count the time you took the observations, and you count the time you uploaded the observations. So, you know, if you don't do it like right away. Any questions on that? Adopt the Loop? Yes.
Just be sure to wear your shirt.
Thank you, Janie, for bringing that up. Janie has reminded me to tell you all, wear your TMN shirt and your badge, because you're going to be in places where maybe someone's going to be looking from overhead, and they'll send somebody to intercept you. My theory on that is, by the time, if they're coming over to us, they're gonna just say hello, because by that time, they've already seen our shirts and they already know that we're there doing a citizen science project. But just in case, do wear them, please, so that you can identify yourselves as a citizen scientist. And there are 2 loops. So the Los Ebanos loop is on San Lucas County Park, National Butterfly Center, Benson, Rio Grande Valley State Park, and Ituria Brush Tract. And Estero Llano loop is Estero Llano Grande State Park, Mercedes Civic Center / HEB Park, Valley Nature Center, and Frontera Audubon. So, you know, it's not just over by Mission area, it's also in Mid Valley and Wesco area where you can volunteer for this. Okay. Give me a sec. I gotta get to the next pick. You know what? I'm gonna bring Logan up. Logan is gonna tell you about a volunteer opportunity and show us. He has some slides to show too. Yay!
Awesome. Alrighty, y'all. Fantastic. So we've been doing this project with the Sherry Heights Garden, and it's coming along really, really well. So let me. There we go. Fantastic. So this is what the garden first looked like when we started it. It's a pretty relatively small little space, as I've mentioned in the past. And so we got to work on that. And here's us out there. This is what it looks like right now. So we took a lot out in the back area. As you can tell there was a lot of buffalo grass here, and we're just kind of slowly working our way in. And here's a pretty exciting part that it's very much in the works, but a very real possibility is extending the garden all the way down to the sidewalk here and eventually expanding out with permission with the city and the golf course, since this is pretty unused space and could be some sort of demonstration garden, some sort of master naturalist project in the near future. But this is very much in the works, but it's an idea that we've thought about and it's very possible. And that is it. So thank y'all.
So the board voted to give $200 to Logan's project to buy plants, flowers for his garden. So you all contributed to this.
By the way, mission text.
It's at the Sherry golf course. Yeah, in Mission.
So if I just put on the little Sherry call numbers.
You could, but if you want, you can contact me or Logan and we'll give you more specific directions. We'll give you an address that you can actually put in. Okay. In your Google Maps. So another project we have is the garden right outside here. Robert, do you want to say anything about that at this time?
No, we haven't scheduled anything, but we do need to clean up the back area.
So Robert says that we do need to clean up the back area, but he hasn't scheduled dates yet. And when he does, he will let everybody know. All right. The other thing is, because it is summertime, we lose a lot of our winter techs and volunteers, and even our permanent resident volunteers, because it's too hot and they won't go out. But our nature areas, our state parks, Estero and Benson, even Resaca de la Palma, and you can even volunteer at Falcon. Our state parks still need help. So if you're interested in earning some hours, you can do something that won't kill you. You can do it early in the morning for an hour, or you can do it indoors somewhere. So check it out. Call those places and find out if there's something you can do. Same thing for our other nature areas like Santa Ana, like Frontera Arban, Valley Nature Center, McAllen Nature Center. All of those places do need volunteers right now. And what you can do is call that place and ask for the volunteer coordinator and ask about what's available that you would be willing to do. All right, especially our trainees from this year. I see some here. That's wonderful. I'm glad you all are here. And get those volunteer hours in so that you can get initially certified. And I think that's it.
All right. I want to tell you a quick story when River said about wear your shirts, because that blimp in the sky, I was told they can see for 5 miles. So anyway, I got to tell you a story what happened to me. I was at Zapata. I had my husband drive me there. Behind the library, there's this, it's right down on the river. I mean, it's all wooded and brush, but they have a cleared area and they have a bird feeder there. So I was looking for seed eaters, which are only up in Stark County and along the river. They like that that habitat, but the grasses there, I don't know what you call that stuff. Those, not really cattails, but something. I don't know. I don't remember the name. But anyway, so I'm there with my binoculars. I'm all alone. My husband stays up in the parking lot behind the library reading. So I went down alone, followed the trail, and I'm near the bird feeders. And I'm looking to see if I see any seed eaters around. All of a sudden, surrounded me were 4 or 5 Border Patrol, all in camouflage, even with the camouflage gaiters up at their eyes with machine guns. And they all at the same time stepped into that circle where I was. It shocked me. I took my binoculars and I went like this, and one of them nodded his head. They stepped backwards and disappeared in the brush, and I never saw where they went. Wanted to give me a heart attack, but someone said you probably disturbed a sensor. They have sensors in the ground, so when people come across over the, across the river, then they set off a sensor and then they come and pick them up. Well, I wasn't going anywhere with them. Oh my God. I talk about it, my heart starts pounding. Okay, I guess that's it. You can claim 0.5 hours for volunteer hour for this meeting. 0.50, and then for the speaker for AT 0.75.
5 for the general meeting plus drive.
Oh yeah, excuse me. I always forget that part. Okay, it's 0.5 plus your drive time. Drive to get here and drive to get home. Don't stop at Dairy Queen. No. Oh yeah. Oh yeah, you're right.
You just take minutes ago.
Yeah. Okay, now I'm going to adjourn the meeting. Our next, the board meeting is July 1st.
Don said we reschedule that to the 8th.
Yeah.
Oh, we did?
Yeah.
Okay, all right.
Because the bank stuff won't be ready yet.
Oh, that's right. Okay, it's going to be July 8th, and then the general meeting will be the next week on the 15th.